I'm really tickled to be able to talk to everyone today, and I hope you're able to get some useful information from the presentation. My degree is in human anatomy, but my area of expertise is histology. I am not a histotechnician, and so I'm leaving all the special stains obviously up to you. But one of the things I do want to talk about is H&E, and in my 30-odd years of teaching histology, I get a lot of questions that are the same over the years. And so I thought if I could relay some of those helpful tips, that may help people be able to identify basic tissues by just using H&E. Now, some of the things we need to think about is, what are the basic features that we're going to be looking for? What are the patterns? And of course, we always have to think about plane of section. So some things to consider when identifying the basic tissues are H&E staining. What's staining? Okay, the plane of section, the resolution of the light microscope. And the importance of that is, will the object or objects even be visible with the light microscope? And then finally, what are the morphological features of the tissue? So a quick overview of hematoxylin and eosin, or H&E, as you all know. Hematoxylin acts like a basic dye, and so it's going to bind to tissue components that have a negative charge, and therefore those tissue components are going to said to be basophilic. They're going to stain blue. Some of the things that we're going to be looking at today are going to be the nuclear staining, and we're going to be looking at glycosaminoglycans, and these are generally repeating disaccharides, and they are sulfated typically, and that sulfation is going to lend itself to the basophilia. Following then hematoxylin, we move into the application of eosin, which we know is an acidic dye, and that's going to bind to tissue components with a positive charge. Anything that stains with eosin is going to be said to be acidophilic or eosinophilic. And some of the things that we want to focus on today, obviously, is the cytoplasm, but I want to focus on collagen and how that's going to appear when we compare some different tissues, and how we want to, what is pink and what should be pink at that point. So plane of section, we all know, is a little bit of a puzzle. We're going from a three-dimensional organ to a two-dimensional slide, and then we have to reinterpret that back to a three-dimensional object that we can imagine as the organ or the cell, that kind of thing. And so we understand too that the plane of section will be determined as we do embedding. And so we have to be mindful, if we're looking at a tubular structure, is it a cross section? Is it going to be a longitudinal section? Is it going to be an oblique section? So again, plane of section is going to be important as we go through. Things may not be visible. Things may look different because of the plane of section. Resolution is something that we need to consider, and that is defined as the shortest distance between two objects in which the two objects can still be distinguished as separate. So the resolution is probably the most important thing for a microscope. And the resolution of the light microscope, as I have here, is 0.2 microns. And again, the resolution is going to be dependent or directly proportional to the wavelength of light. And so that's going to pretty much limit what our resolution can be. So the question we have to ask ourselves is the size of the object at or below the resolution of the microscope. So if it's at the resolution, we might see it. It's not going to be clear, but if it's below the resolution, we definitely aren't going to see it. So I went ahead and put 4 common structures listed here with their sizes to give us a little bit of a reference here. So the plasma membrane, or the plasma lemma, the outer cell membrane, is somewhere between 7.5 and 10 nanometers. The ribosome is being much larger, 20 by 30 nanometers, and the mitochondrion is huge, a diameter of basically 0.5 to 1. But if I look at that length, it can be really quite excessive or extensive. And then, of course, the nucleus is the largest of our organelles, and that can be up to or beyond 6 microns. So clearly, of this 4 lists, of these items in this list, 2 are going to be visible with the light microscope. So the object of this presentation is to review the 4 basic tissues, talk about some of their characteristics, and I want to go ahead and maybe address some of the potential histology lookalikes. And I want to end our presentation today by giving you a small quiz and see if you can identify what the potential histology lookalike is. So our 4 basic tissues are listed here: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. So epithelia typically forms sheets of cells that cover surfaces, lines cavities, hollow organs, and the function is going to vary depending on where it's located. So covering or surfacing epithelium is always going to be at some kind of an interface, and that interface could be, you could have urine if you're in the urinary bladder, it could be air, so it could be contents of the stomach. So the function then is going to depend on the location of that epithelium. Also, we want to remember that epithelial tissue forms glands, and glands basically are epithelial cells that make and secrete a product. Next up, we have connective tissue, and these function in support, structure, and defense. Third, we have muscle tissue, which is contractile, which means it shortens, and therefore it allows for movement. Nervous tissue is conductive, and it basically integrates. So now with that basic introduction, let's consider how we're going to try to identify these 4 basic tissue types. So before each of our tissues, I'm going to highlight the tissue that I'm going to talk about. So we're going to talk about epithelial tissue. And one of the things I want to point out is that it's very cellular and it has polarity. And we'll define what that is here in just a minute. So now for looking at this particular feature here, and I want to point out my source here. This is Jonquier's Basic Histology, Text and Atlas, and it just had its 50-year anniversary. I used this book back in the 80s, and it is really, really great. And Anthony Mesher and McGraw-Hill were most happy to have me use this during the presentation. So all of the figures are from Jonquier's Basic Histology, and most of the micrographs, unless they're indicated, have been taken by myself. So let's look at some of these epithelial features. So first of all, if you follow the arrow, it's very cellular. So the cells are close together, okay? Which means we're going to have little extracellular matrix. In that sense, we're going to have then specialized cell junctions holding these cells together, okay? We also say that epithelium is avascular. So we have no blood vessels running through the tissue. Down here is going to be a gland. And again, we don't have vessels running through that epithelium. If we notice here off to the side, we have a capillary or a small vessel. And over here, we have a small vessel. So even though epithelium is avascularized, it always has a good source of epithelium with it. One of the things that we want to point out is that the epithelium is always associated with a structure called a basal lamina. And that basal lamina is a non-cellular component. It's a sheet of type 4 collagen, glycoproteins, etc. And it is always present. So we have epithelium that rests on that basal lamina. Sometimes we refer to it as the basement membrane. We get a little lazy with that, but understand that the basement membrane basically has 2 parts. It has a basal lamina and a reticular lamina. And generally we say if you can see it with the light microscope, people call it a basement membrane. So let's go to this last point, polarity. Polarity is only demonstrated when we talk about epithelial tissue, meaning it has 2 distinct surfaces. So we have the surface that's down here that rests on the basal lamina, and then we have the surface up here that is free. So we have the uppermost, which we're going to say the apical, and then down here we're going to have the basal. We also then have a lateral surface on either side. If we look at this then, we have distinct proteins, and we have unique features of the membranes at the apical as well as the basal area. And so we need to pay attention to that polarity. Again, this is the only tissue that has polarity. Associated with all epithelium is a connective tissue, and that's called areolar or loose connective tissue. It is full of numerous blood vessels. It has many defensive cells, and we'll go ahead and talk about that in a few minutes. So general features of epithelium. Now, one of the things that anatomists like to do is name things or classify things. So let's see what's happening with that. So if we look at the classification of epithelium, you know that we like to count and we like to look at shapes. So first thing we have to do is how many layers of cells do we have? If we have 1 layer of cells, we're going to say it's a simple epithelium. If we have 2 or more layers of cells, we're going to have a stratified epithelium. We then go ahead, and if it's a simple epithelium, all the cells should basically have the same morphology. There will be an exception. So if we're looking at stratified epithelium, we're going to look at the most apical layer, and I'm going to show you that in just a minute. Our three shapes are as follows. The cells can be squamous, flat, cuboidal or cube-shaped, and finally columnar, which means they're taller than they are wide. And so what happens is, because we cannot see the cell membranes, what we really want to do is look at the morphology of the nucleus. If the nucleus is basically flat, it's going to be squamous. If it's round and in the middle, we're going to call it cuboidal. And if the nucleus is either oval or round, it's going to have a tendency to be at the bottom or the basal aspect of the cell. So again, the position of the nucleus is going to be important. So this is a really nice chart taken from the textbook. And one of the things I want to point out, if we have two options of simple epithelia versus stratified epithelium, it makes sense then we have simple squamous, simple cuboidal, and simple columnar. Also, it makes sense that we have stratified squamous, stratified cuboidal, and stratified columnar. But we can see that there are some variations of that. So let's take a look at this. Of all the epithelia, simple squamous is going to be the thinnest, so it's going to be great for diffusion. Simple cuboidal and columnar are going to be great for secretion and absorption. And then if I come down here to the very bottom where it says pseudostratified, pseudostratified is a type of simple epithelium, meaning it looks stratified, but it's made up of one layer of cells, which means we have cells that are short, tall, and medium, so it has a stratified appearance, but is classified as a simple epithelium, and we'll see examples of that here shortly. Looking at the stratified epithelium, it's going to have two or more layers. If we look at stratified squamous epithelium, this is the thickest of all the epithelia, generally provides some good protection from abrasion. If we look at the two different types, we see the keratinized, which is going to be dry. We see it in the epidermis of the skin. And we see the non-keratinized, which is going to be moist, which we're going to see lining the mouth, oral cavity, the esophagus, etc. Morphologically, the difference between these two is that in stratified squamous, non-keratinized, you're going to see epithelial nuclei all the way to the very most apical surface. So all of the cells have a nucleus. These cells are all viable. When we talk about stratified squamous keratinized epithelium, the upper layers are too far away from the nutrients. They're dying, they're dead, basically. Their nucleus is going to be autolysed or consumed, and so we will not see nuclei in the most apical layers. We're not going to look at skin, however, today. So we also then have stratified cuboidal and stratified columnar, which have very small distribution. But one of the things I want to point out is transitional epithelium. This is kind of an unusual one. This is found lining the hollow organs of the urinary tract. And so if we look at some of these possibilities of simple and stratified epithelia, we can have some lookalikes. So I wanted to go ahead and see if we could figure out what we're looking at here in the next slide. So if I look to the left over here, this is stratified squamous non-keratinized epithelium. I go right to the surface and I see the presence of nuclei. So I know that this is non-keratinized. I go down here now to the basal layer. This is the basement membrane down here. So this is the basal layer. This is where the regenerative cells are going to be. It doesn't matter what the shape of these cells are down here. We want to look at the apical layer. So this then is classified as stratified squamous non-keratinized epithelium. We're going to come back to this one in just a minute. If I look over here, I see these roundish profiles. And if I look at these, I have to say, well, it has polarity, but let's make sure we understand. On the outside of these ring-like structures is where the basal lamina would be, and then this is where the apical aspect is going to be. If I look at these then, I see a round nucleus, and it pretty much occupies the middle of the cell. This is classic simple cuboidal epithelium. So the bulk of this is simple cuboidal epithelium, and this is taken from the kidney. Now, as we look around, you go, wait, here's a cell that doesn't have a nucleus. Over here's a cell that I don't see the nucleus. And so we're not alarmed by that because of the plane of section. So we need to be mindful of that. In a plane of section, we may not see all the cellular detail that we would like to. So now this is a common problem I have with my students. They want to confuse these two types of epithelia. So on the left, we have the stratified squamous non-keratinized epithelium. Again, I refer you to the apical surface where the nuclei are going to be flat. It is definitely stratified. This is a nice thick epithelium. If I come over here to this side down here, and the transitional epithelium, this is where the basement membrane or the basal lamina would be. This is the apical surface up here. One of the things I want to bring your attention to are these large cells up here on the surface, these cells up here on the surface, and you know these, of course, as dome cells or umbrella cells. Sometimes students will confuse this with stratified squamous or stratified cuboidal epithelium. But one of the things I want to point out is if we look, transitional epithelium in a relaxed, undistended state should have approximately 6 layers of cells. When it's distended, these dome cells are not going to be visible. So we're going to stretch from 6 layers to approximately 3 layers. And so then the question is, am I going to be able to distinguish distended transitional epithelium and stratified squamous non-keratinized epithelium? And I refer you then back to the overall thickness of the stratified squamous non-keratinized epithelium. So these are two potential lookalikes, keeping in mind that transitional epithelium has these unique dome cells that we can clearly see when the bladder or the ureter or the urethra is non-distended when it's relaxed. So now we have some potential problems here. So I'm looking at two types of columnar epithelium. On the left, I have what we call pseudo-stratified columnar. So if I look at this, it looks like the epithelia, or excuse me, the nuclei are at different heights. And indeed, they are at different heights. Okay, so we have some that are low, some that are in the middle, some that are high. So what happens is, this is basically found in the trachea. So this would be a respiratory type of epithelium. There are 5 types of cells that we can identify in this type of epithelium. But the thing here we want to understand is all of the cells are resting on the basement membrane. So we have short cells, medium cells, and tall cells. We're going to come back to this type of epithelium in just a minute. Over here on the right, we have something called simple columnar epithelium. And if I look at this, the nuclei have a tendency to appear in the same row. So it's a little more organized. You see a little more distinctive cytoplasm up here, whereas opposed to the other one, it looks a lot more unorganized. I want to spend a little time on these two because these can be problematic. So this picture is taken from Jonquière text, and I want to point out some things here. If we look at this presentation, which has a very nice simple columnar epithelium, you can clearly see that these nuclei are oval and they are lined up. So I can kind of draw a nice line right through those, okay? When I come over here and look at the pseudostratified columnar epithelium, again, I notice that the epithelium are at all different levels, okay? Now, one of the things you may ask yourself are, what are these guys? What are these round cells that are indicated by the white arrow? Those are lymphocytes that are now migrating through. So these nuclei, the elongated ones, are belonging to our epithelium. Now I want to go ahead and look at these two epithelia again, looking at surface features. So let's take a look at this. There are some special considerations that we need to think about when we're looking at epithelium. And so if I consider the apical aspect, these are some of my options. I can have cilia, which are very long extensions. These can be up to 10 microns in length. So 5 to 10 microns in length and about 0.2 microns in width. So these are easily seen with the microscope. They're full of microtubules that are organized and allow for the cilia to move material across the surface. If I come over here then on the right-hand panel, I see these short little structures right here. These are microvilli, and they're roughly 1 micron in length and about 0.1 micron in diameter. So there you can see them, but you're going to have to work a little harder at as far as the resolution goes. If we have a lot of microvilli, these are often called a brush border, or it's often sometimes called a striated border, depending on where we are. Microvilli are not unusual in epithelial cells, so we generally don't identify them when we're talking about epithelium. What we do want to indicate is the presence of cilia. So I want to take just a moment and talk about cilia and the cell that's indicated right here, the goblet cell. There are only two systems in the body that have cilia. Those are the respiratory system and the reproductive systems, both male and female. Again, there's only two systems that have cilia. The digestive tract, the digestive system does not have cilia. Material is moving along via peristalsis of the smooth muscle. So again, if you see cilia, that's going to narrow down where we could be. Now, regarding goblet cells are basically unicellular exocrine glands that produce mucus. So these are going to be lovely with PAS stain, as you know. What happens then is goblet cells are only found in two systems. They're found in the respiratory system and they're found in the digestive system. So they're only found in the large and small intestine. So again, if you see some of these features, such as cilia in the goblet cell, that's going to indicate, kind of narrow down where we are in the body. Now, if the pathologist gives you a specimen, you don't have to really guess where it's from. But if you're teaching a histology class and you're trying to identify tissues, some of these are good things to know, especially when you're trying to figure out what system it belongs to. Now, I also want to just briefly mention glandular epithelium. So the goblet cell was an example of a unicellular exocrine gland. And here, what we see is a multicellular exocrine gland. And these are mucus secreting, and they're delightful, okay? And so what happens is I can identify these because we have the flattened nucleus at the base, and we have the lighter staining mucus up here. So you have exocrine glands and you have endocrine glands, and they are defined based on the location of their secretion. So an exocrine gland, by definition, is going to release its product onto the surface of an epithelium. Endocrine glands are going to release their product into the extracellular matrix or into the blood. So we're going to step away now from epithelium, and we're going to move on to our next tissue type. And that is connective tissue. The thing with connective tissue that we want to spend time on is that this is different from the other types of tissue because the thing that we like to look at is the extracellular matrix. It's the predominant component, or it's the prominent matrix that we want to be particularly mindful of. So let's consider the components of connective tissue. We have two basic things. We have the extracellular matrix, and of course we have cells. So let's look at what we see in the matrix. We have fibers and we have something called ground substance. So fibers are going to be basically fibular or long, thin proteins. They come in the form of collagen, reticular, and elastic fibers. And we'll talk about staining with those as well. Ground substance is more like a jelly-like material. It is made up of proteins and carbohydrates, and specifically the PGs here are proteoglycans, which are going to have a core protein with a bunch of glycosaminoglycans. It looks like a bottle brush. Then we have the GAGs, GAGs, or glycosaminoglycans, and these are made up of repeating disaccharides, and they're mostly sulfated. So we're going to have a negative charge, which means if they're properly prepared, they will stain with hematoxylin. They're going to be basophilic. And then over here we have glycoproteins, which are predominantly proteins with a little bit of carbohydrate on them, and things like fibronectin and laminin. And then finally, a component we have to address is something called the interstitial fluid. So the interstitial fluid is going to basically be water and such, and it's going to be held in position, if you will, by the electrical charge, the attraction of those sulfated glycosaminoglycans. Cells that we're going to look at are fibroblasts that make the matrix, mast cells, plasma cells, macrophages, the white blood cells, the adipocytes, and then we get into some that we see in the specialized connective tissue, and those are the chondrocytes and the osteocytes. This clearly is not an exhaustive list. So we like to classify things. That's what anatomists do. So we're going to kind of group them into two categories. So we have the basic or proper connective tissue. And what we want to say here is this is called basic because the extracellular matrix is not specialized. If we come down here, then we also then have the specialized connective tissue where the matrix is unique. So if I go back now to the basic, I see we have two different varieties, loose versus dense. The difference between these is that dense connective tissue has an abundance of fibers at the expense of the ground substance and cells. So the predominant component in dense connective tissue is going to be the fibers. Loose connective tissue, we're going to see lots of ground substance, we're going to have fewer fibers, and we're going to have more cells and more variety of cells. And we'll see examples of these. The specialized connective tissues you know as cartilage, bone, and blood. We are not going to look at blood, but we're going to take a look at cartilage and bone. So here we are looking at a loose connective tissue. We have three types: areolar, adipose, and reticular. Areolar loose connective tissue, as we see right here on the bottom, is the most widespread, has the widest distribution. It has the most variety of material. It has all three fiber types. It has all of the ground substance, and it has an incredible diversity of cells. This is associated with all the epithelium, and it surrounds blood vessels, and it surrounds nerves. And this is taken from a mesenteric spread. On the left, we have adipose. And so this is a collection of adipocytes, and then each one of these is going to be an adipocyte. And we know in preparation that the lipid is removed. And if we want to see that, we're going to have to use things like oil red O or Sudan black or even osmium tetroxide. Over here, I want to move over then to the reticular connective tissue. So reticular connective tissue is made up predominantly of reticular fibers. Reticular fibers are made up of type 3 collagen with roughly 10% carbohydrate, which is going to be different from collagen, which is only going to have a 1% carbohydrate. So these reticular fibers then are going to respond to special stains, such as Gordon and Sweet silver stain. It's going to also respond to PAS. So this is taken from the liver, and this is using Gordon and Sweet, and these are the reticular fibers. So again, we're not going to see these reticular fibers with standard H&E. If I'm looking then at dense connective tissue, typically we think about collagen. The most abundant protein in our body is the collagen. And so when I think of dense regular and dense irregular connective tissue, I'm referring to the organization of the collagen. We also have a type of dense connective tissue called elastic connective tissue, and the predominant component is going to be elastic membranes and elastic fibers. So over here in the figure, we're going to have an image of dense irregular connective tissue. This is taken from the dermis of the skin. The eosinophilic structures in here, this is all the collagen fibers, and you note they're going in all different directions. The nuclei that we see are going to be from the fibroblasts. And so basically the point of dense irregular connective tissue is that it's going to be able to take pulling forces from all different directions. Next up, I want to compare dense irregular connective tissue with H&E and with special stain. So on the left-hand side, I have dense irregular connective tissue. This is from the mammary gland. And again, this is stained with H&E. We can see the nice, rich collagen fibers going in all different directions. Over here, we see the same tissue, but it's stained with Masson's trichrome using aniline blue. So we see all the nice fibers in here, and the nuclei typically belong to fibroblasts. If we now go ahead and look at two other types of the connective tissue, on the left-hand side, we have dense regular connective tissue. This is collagen fiber that's arranged in layers, so it's organized. This is what we find in tendons and in ligaments. This is going to pretty much resist pulling forces from one direction. And again, the eosinophilic is the collagen fibers, and the nuclei are going to belong primarily to fibrocytes in this case. If I come over here on the right-hand side, this is taken from the wall of an aorta. This is elastic connective tissue. So these wavy things are elastic lamina. Very interesting. And you can actually see those with H&E. If we adjust the iris diaphragm, if we adjust the condenser, we can make elastic membranes and fibers more refractile. So let's take a look at this. On the left-hand side, we have then H&E staining of the human aorta, and we can see then the waviness over here. And then over here, we have with Verhoef von Giesen, so we have an elastic stain, but you can see that this clearly has a different morphology from what we saw with our dense regular connective tissue. So let us now look at this. If I come over here, I see specialized connective tissue, and I want to move now into cartilage. So over here on the left-hand side, I have hyaline cartilage. On the right-hand side, I have elastic cartilage. The cell that we see here is called the chondrocyte. The chondrocyte is a roundish cell that sits in a little cavity, and that cavity is called a lacuna. When the cell undergoes mitotic activity, it forms little clusters. Those little clusters are called isogenous nests or isogenous groups. We will not see these clusters of cells in bone. When I look at hyaline cartilage, it's said to have an amorphous matrix, which means it's smooth. We do not see any fibers. The basophilic material that I see around the periphery is going to be the sulfated proteoglycans that are going to be basophilic. So this is a nice preparation of hyaline cartilage. I come over here now and look at elastic cartilage. And I go, okay, I still see the chondrocytes. I see them in clusters, isogenous nests. But now I see these fibers. This is with H&E, and these fibers are elastic fibers. And again, if you adjust your iris diaphragm, your condenser, you can actually see that these are refractile. But typically, these are identified when they're stained with elastic stain. So here we have, in comparison, elastic cartilage with H&E, and we have the elastic cartilage with Verhoff von Giesen. And again, clearly we can see the elastic fibers here by the stain, and here we know they're there based on the refractile activity. The cellularity of elastic cartilage and hyaline cartilage is going to be similar. Now, the elastic fibers are going to give the elastic cartilage some flexibility. So we're going to find this tissue in our outer ear, in the pinna of the ear. So we can go ahead and bend that around and around, and it's not going to break. So that's going to be nice. We then have two other comparisons to make here. And again, these are commonly confused, although let's find out why they shouldn't be. On the left, we have fibrocartilage. Fibrocartilage is one of those that's kind of a hybrid between hyaline cartilage and between dense regular connective tissue. The fibers that I see over here are the collagen fibers, the type 1 collagen fibers that we saw on the dermis of the skin that we see in dense regular and irregular connective tissue. But I look over here and I have little pockets of chondrocytes. And these chondrocytes are producing this basophilic cartilage matrix. So we have a hybrid, if you will. And these are found, this is from an intervertebral disc, a human intervertebral disc. And if we note, there's not many cells in this particular tissue compared to the other types of cartilage that we saw. A lot of times in fibrocartilage, the cells are going to be single, or they're in maybe a small cluster or maybe rows. So I thought this was a nice presentation. Over here again, we're comparing this with elastic fibers or elastic cartilage. Again, I see these fibers, but I know that these are elastic fibers, whereas in the other one, those are collagen fibers. Note the difference in the cellularity. Elastic cartilage is very cellular compared to fibrocartilage. Fibrocartilage in the intervertebral disc can take the compression based on the hyaline aspect, and it's able to resist pulling forces based on the collagen. So it's a nice compromise between those two tissues. Now let's look at the last type of specialized connective tissue, and that is bone. So on the left-hand side, we see compact bone, which has been decalcified and stained with H&E. On the right-hand side, we see the compact bone that's been in a ground bone presentation. And again, we see this is commonly what we see. We see the round structures here. So we see the osteons, the functional unit. In the center, we're going to see the Haversian canals. And of course, we're going to see the Volkmann's canals right here. I can see these same structures over here. I can see the Volkmann's canals over here and over here. I can see the central Haversian canal, and I can make out that round target that I saw that is the osteon or the Haversian system. What we see, however, in the decalcified bone is going to be the organic matrix component. And this means 90% of our organic matrix component is going to be collagen type 1. And we know that collagen is going to stain with eosin. And so this is why we have such a very nice eosinophilic appearance here. Compact bone, we don't have any stain. So now I have 3 slides in here to see what we've learned, okay? I want to start in the middle, and I put this one in. This is a bad slide. And you look at it, it's bad because poor preparation. This tissue is not well fixed. This is hyaline cartilage. And if you look at this, you have no indication that there's any kind of proteoglycans here. This is basically all eosinophilic. And this is what a lot of our slides have a tendency to look like when you look at them over time and you go, how can this be hyaline cartilage? But this is why I picked it. If I look at this, I see the chondrocytes again, they're going to be round cells. They shrink in fixation, but I also see clusters. So I see these little isogenous nests. So to me, this is always going to be a cartilage, and I don't see the fiber. So I'm going to have to go to hyaline cartilage on this. If I go to the left over here, I see pink again. Well, this is type 1 collagen. This is actually bone. This is lamellar bone. And each one of these little cells right there is an osteocyte. So in bone, you will not have isogenous nests of cells. These cells will not undergo mitosis. And the lacuna where the cell body sits is going to be oval. It's going to be oval. Well, I want to point out that bone is the only connective tissue in which we have cell junctions. So we have gap junctions that are connecting this cell to this cell. So that's unusual. The reason we have to have gap junctions is we're not going to have diffusion of nutrients through the hard matrix. So again, on the left-hand side, we have bone, which is going to be eosinophilic and it's decalcified bone. It's going to be eosinophilic because of the collagen. We have single cells, osteocytes. In the middle, we're going to have hyaline cartilage that was poorly prepared. Very cellular, but we see nice isogenous nests. So that tells us that this is hyaline cartilage. And then I come over here. This is also collagen, but this is dense, regular connective tissue. And so what we want to do now is this particular type of tissue going to be confused with anything else? So we're going to move along to see if there's more potential histology lookalikes. So this is a wrap-up of our connective tissue. So now let's take a look at the nuclear morphology. How can we identify some of the different cells? So very quickly, I would like to say the following. We don't always see the cellular detail that we would like to, so we want to kind of pay attention to nuclear morphology and cytoplasmic detail. For example, the fibroblast or fibrocyte has an oval nucleus. The cytoplasm typically is not obvious. The plasma cell, which is going to have a round nucleus, has abundant basophilic cytoplasm. That's going to be really obvious. This cell is busy making antibodies, so it's going to have a lot of rough endoplasmic reticulum. The fibroblast is going to be making all the extracellular matrix that we're going to see in the general connective tissue. The lymphocyte, which is an immune cell, in section is going to basically look like a round, naked nucleus. No visible cytoplasm with H&E. So we're going to have to say, how can I now distinguish between these three cells? I'm looking at the morphology of the nucleus, and I'm also going to say, do I see cytoplasm and how's it staining? And lastly, in this group, I included the mast cell. The mast cell also has a round nucleus, but unlike the other ones, it has obvious granules. And so the mast cell is involved in allergic reactions, and so it's going to have histamine and heparin, which is going to allow those blood vessels to dilate and become leaky. And so they're going to be able to be seen with special preparation. Finally, I included these other two cells that could be seen together. If we're looking at a bone marrow, if we're looking at a section, a decalcified section, we may see these two large cells at the same time. First of all, we have the osteoclast, which we see in bone tissue, multinucleated with eosinophilic cytoplasm. So this is used to remove bone tissue. The megakaryocyte is another large cell that we can see in the vicinity. And it's going to have a large polyploid nucleus. So it's going to be a large nucleus, but it's going to be all continuous. So we're not going to have individual nuclei. And again, we're going to have eosinophilic cytoplasm. So we're going to see if we can figure those out. And the reason they may appear close to each other is because in preparation, those osteoclasts may be pulled off of the surface of the bone. So if I look at this, this looks like a mess. This is a lot of connective tissue cells, and let's see what we might see. I'm going to come over here and see these large kind of void cells. These are nuclei of the macrophage right here, okay? I come over here, I see a round nucleus, and I see lots of cytoplasm off to the side. I go, hey, that looks like a plasma cell. So we have plasma cells. And so we need to say, what is the morphology? Is there cytoplasm that's visible? We're going to come back to some of these examples shortly. Now, I like to do a comparison of these two types of cells side by side. The lymphocytes on the left versus the plasma cell on the right. The lymphocyte, we have a bunch of round nuclei, and again, we don't see any obvious cytoplasm. And again, you do when you look at a peripheral blood smear or blood prep, but in a section, we are not going to see that cytoplasm. If I look over here at the plasma cell, and this little guy's isolated, but really nice, we have this round nucleus. It's typically eccentric, so off to the side. We often say it has a clock face nucleus, so you see like 12 o'clock, 1 o'clock, and we go around. And then look at this abundant cytoplasm. The cytoplasm is very basophilic, and it's basophilic because of the abundance of rough endoplasmic reticulum. If you look at this area right here, you see where it's a little light. That light area is called the HOF, H-O-F. You don't hear that term too much anymore, but this is where the Golgi and the centrioles are going to be located. So Golgi and centrioles are not going to stain with H&E, so you're going to have a lighter appearance where they are. So again, we're looking at two cells with a round nucleus, but again, do we see cytoplasm? That's going to be the key. Okay, so let's take a look at this. So now we have a whole bunch of plasma cells here. Once you see one, they just start popping out all over the place. And notice that they may not be perfectly off to the side. Here's a plasma cell, there's a plasma cell, there's a plasma cell. So we have lots of cytoplasm, but it may not be perfectly off-centered like it was in the previous slide. Now, the large two cells that I wanted to take a look at is, if I look over here on the left, this is the osteoclast. I count 1, 2, 3, 4 nuclei in this field right there. This is the bone surface right here, the matrix. So this would be what we would call the Halschip's lacuna or the resorption bay right here. This is where the matrix was removed, okay? But notice in preparation that that osteoclast can be slightly pulled away from the surface of the bone. Over here on the right-hand side, we have this large cell. This is the megakaryocyte, and it has a polyploid nucleus. This cell is going to give rise to platelets. And if we look around here, we see cells that are undergoing hematopoiesis. So again, we can have areas where we're going to have bone remodeling, where we're going to see an osteoclast, and maybe not too far away, we're going to see a megakaryocyte. Megakaryocytes can be easily seen at 4x because they're so big. But again, separate nuclei on the osteoclast, one giant nucleus in the megakaryocyte. Okay, let's go to muscle tissue. So muscle tissue is very cellular and it has no polarity. Let's take a look. We have three types of muscle tissue, as you know: skeletal, cardiac, and smooth. Features include very cellular, it's excitable, it contracts, and we have two kinds of classifications based on the morphology. They're either striated or they are unstriated. Let's take a look. This is a very nice figure. So on the left here, we have skeletal muscle. We see obvious striations. This tells me that the myofilaments, actin and myosin, are organized into sarcomeres. And it's those sarcomeres that are going to cause this banding. So the AI band, we have nice banding here. And we're going to have multiple peripheral nuclei based on development. So one of these long things is a muscle fiber, a muscle cell. And we can see then the drawing down here that coincides with that. Very nice. In the middle, we have cardiac muscle. We have striations that are visible, but we have a round central nucleus. We may have two, but where the two ends of the cells meet, we're going to have a structure called the intercalated disc. These cells are branched. These cells are branched. And this is one of the tissues, I think, that's probably one of the hardest to try to identify in section. The organization of cardiac muscle, because of the way it twists, you see often cross and longitudinal section, and it's kind of tricky to see when you see a lot of cross section. The last smooth muscle type, or excuse me, the last muscle is smooth muscle. These look like little kayaks, or they say they're fusiform. So we have an oblong shape and the central nucleus. And so these cells are nicely packed together, and we can see the rendition down here. Now notice again, we're not seeing a nucleus in every cell because of plane of section. This, I really like this figure, and I wanted to include this. If you look, this goes back to the 12th edition. So this is an older picture that they had. But I like this because it shows the longitudinal versus the cross section. We know what the longitudinal looks like in the sense we're going to see the striations, the intercalated discs, et cetera, et cetera. Let's pay attention now quickly to the cross section. If I look at skeletal muscle and cross section, I'm going to see the peripheral nuclei. Generally, they're going to have the same shape, but they may have a little different size. If I look down here now at cardiac muscle, this is where things get a little tricky. If you recall that cardiac muscle is branched, so when we cut through it, we're going to have areas that may be two-sided. There's going to be one that may be a smaller aspect. You may see a nucleus. You may not see a nucleus. This is a very odd-shaped tissue in cross-section. And then finally, I'm looking at smooth muscle. And again, depending on the plane of section, I can either get the center of the cell or I can get the ends of the cells. So I may or may not see a nucleus, but the profiles will generally be round, but they're going to be different sizes. So here we have H&E of skeletal muscle, longitudinal and cross section. We have a little separation here, but that's okay. We have cardiac muscle, so we have longitudinal over here. We can see the fibers that are branching, and then over here you can see this is kind of a mess over here. This is classic cardiac muscle. And then smooth muscle, this is nice. And if you look, you see a little bit of a wavy appearance in the nucleus right here. It looks like almost like an inchworm. That tells us that this muscle is contracted, okay? But notice how dense the nuclei are. So we have, this is very cellular. Notice the cross section. We have pretty much round profiles. They're going to be of different sizes. They may or may not contain part of the nucleus. So now the question is what is this? What type of muscle tissue is this? So if I go along here, this is fairly longitudinal right in here. This is cross section. This is kind of cross section. And I go, oh, I see intercalated discs. I see intercalated disc. I see intercalated disc. So this is cardiac muscle. So again, it's really hard to just get pure longitudinal cardiac muscle based on the anatomical arrangement in the myocardium. So now we move back to the last tissue type, and that's nervous tissue. And nervous tissue I threw in just for completeness, but we want to say that it's very cellular, but it doesn't have polarity. So let's take a look. The two basic types of cells are going to be neurons and the neuroglial cells, or the supporting cells. This tissue is very cellular, but unlike epithelium, the cells don't form sheets, they're all intertwined. The neurons are excitable and communicate via synapses. And the neuroglial cells are going to be supportive. So they're going to be involved in ionic balance. We're going to have macrophages, et cetera, et cetera. So they're really, really important. Okay, so let's take a look at this. When someone draws a neuron, this is typically what they draw. It's called a multipolar neuron. So we have the cell body, and then we have all the extensions that come off of it. This then is the cell body. This is where the neurotransmitters are made. The bulk of these extensions are dendrites, and then we have one long axon. So the reason people draw multipolar neurons is these are the most common type. Interneurons, which are housed entirely in the CNS, and motor neurons are all multipolar neurons. And so this is really, really the most typical type of morphology that we're going to find. So the reason I'm bringing this up is I want to draw our attention to two things, the cell body and the axon. Depending on how we want to break down the tissue, how we want to organize it, if we're in the CNS, we're going to say that we're going to have central nervous system, we're going to have the brain and spinal cord. The tissue, as you know, is going to be organized into gray and white matter. We don't say gray and white matter when we're talking about nerves or ganglia. So let's take a look at this. This is one of those multipolar neurons. Here's one here, and here's one here, here's one here. So we are in the gray matter of the CNS. This thing right here is an axon. This is the axon hillock. It was a really lucky section, okay? And I know that this is the axon because it does not contain Nissl. And so all of these little speckles in here are Nissl. All of these other nuclei out here belong to the supporting cells of the neuroglial cell. So generally, we're pretty good at identifying nervous tissue and identifying neurons. But let's take a look at nerve. So if I say nerve, if I say peripheral nerve, it's kind of redundant because if one uses the term nerve, that means we're out in the periphery. So I want to take a look at this with higher magnification and then pop out here to lower magnification. If I look at this, most of our nerves are myelinated, highly myelinated. And so if you follow my arrow, we're going down the axon, okay? This white space out here is the myelin that was washed away in tissue processing, So we have this vacuolated appearance. This pink stuff that you see, it almost looks like herringbone. Those are called our Schmidt-Lederman clefts. That's basically the cytoplasm that got squished out in the process of myelination. Here's a nice little node of Ranvier if anyone wants, they're nice, okay? The main thing I want to point out is all of this is lipid and all of this is going to be washed out unless we use osmium tetroxide. Let's come over here and look at this. Under lower magnification, could this be confused with any other tissue? I want you to focus on how light these areas are. This lightness right here indicates that the lipid has been removed. So nerve in longitudinal section almost looks bubbly or vacuolated, okay? So I want to compare nerve with smooth muscle. That seems to be a good histology lookalike. So again, with tissue preparation, the myelin is removed. And so we have this light vacuolated or holy appearance. So here we are, histology lookalikes. What are they? Well, we just saw this one before, and I noticed again the white space is here, so I know this has to be nerve. And I go here and I go, oh yeah, there's a node of Ranvier, and this is actually quite nice. I want you to compare the intensity of these two. The one on the left, the eosin is fairly strong. Notice that we don't see the vacuolation. It doesn't look bubbly. This is smooth muscle. And if we look at smooth muscle, we see then that it's very cellular. The cells are packed together, so we see lots of nuclei. So again, we have some difference in these two. But again, looking at the lipid removal is going to be a key feature for identifying the peripheral nerve. Now it's time for a quiz. So as a professor, one of the things I have to do is quiz people. And so you're in luck. I have 8 questions for you. Here we go. Which tissue types are present on this slide? So again, everything's going to be H&E. I want you to focus down here versus up here. Up here and down here. So what 2 tissues do I see? Well, this one's going to be more intense. This is very eosinophilic compared to this one. If I look up here, the nuclei, there are a lot of nuclei, okay? Lots of nuclei. Down here, here's a nucleus, there's a nucleus. This is dense, irregular connective tissue, and this is smooth muscle. So I like to tell people when they're looking at this, if you're looking at smooth muscle, your ratio of hematoxylin to eosin is maybe like 1 to 4. If I'm looking at dense connective tissue, my ratio of hematoxylin to eosin is maybe 1 to 15 or 1 to 20. So you're going to have a lot more eosin, the collagen fibers, compared to the nuclei. What's this? Well, I see all things going in all different directions. So is this smooth muscle? No, it's vacuolated. So this actually is a nerve that's actually kind of cut in a funny plane. That's an axon. That's the myelin that's been removed. So again, you see roundish profiles, but it is the void of the material in there. It's the lipid that was removed. So that's how I know that that is nerve. All right, what's this one? Well, I see pink. I don't know what that pink is. Okay, actually I know what that is, but I'm looking at these long things. And I go, what are these? These are extensions. So these are multipolar neurons, and so this is nervous tissue. So this is basically from an ox spine smear. So this is a smear of nervous tissue, unlike the one we saw earlier, which was the section of the gray matter. But because I'm seeing neuron cell bodies, I know this is gray matter. Aha! What cell are we seeing in this connective tissue with the red arrow? It is, again, the plasma cell. It's my favorite cell. That's why you see it again. Sorry about that. Now, here's a good question. Are these structures right here bands of smooth muscle, or are they nerve? What you think about that? You see lots of nuclei. It's not overly clear. We're at a distance now. So what's it going to be? What do you think? Well, one of the things I want to draw your attention to is if I come out to the periphery, I see a connective tissue on the outside, okay? This is perineurium. So this is a connective tissue wrap that's going to surround the nerve. So this is nerve and this is nerve. When you see smooth muscle out in an organ like this, you're not going to see that band of connective tissue around it. So this is nerve. We're now in aerial or loose connective tissue. Do you see different types of cells? So I see cells with granules. These are going to be the mast cells. I see cells that have a ghost-like nucleus. This is going to be the macrophage. I see potential lymphocytes, okay? I see fibroblasts. I see elastic fibers. I see all kinds of good things in there. This tissue is what? So let's look at it. I see lots of vessels. I see lots of ground substance. I see lots of nuclei, lots of cells. So this is going to be aerial or loose connective tissue. And finally, the question is, what tissue is this versus what tissue is this? Again, this is smooth muscle. And the collagen now is more of a less pink. So the different subtleties in the staining, you can actually nicely tell smooth muscle from connective tissue. So special thanks to Anthony Mesher and McGraw-Hill for the use of the book. And I'd like to thank you for your attention. And if you have any questions, you can always follow up with me at my contact information